The spinal cord communicates through axons, so regeneration, or stimulating the regrowth of axons, is an essential piece of the puzzle in finding a cure for spinal cord injury. The problem, though, is that spinal cord injuries result in neurons dying, whether it be a result of the initial traumatic event or the damage from the secondary phase of injury. The injury also triggers the formation of the glial scar and release of molecules that prevent the neurons from regenerating. Since the central nervous system needs neurons to communicate and neurons cannot grow back or regenerate on their own, two things need to happen. First, we need to break down the glial scar and decrease the amount of inhibitory molecules that surround the site of injury. And second, we need to increase axon growth and remyelinate damaged axons. Inhibitory molecules like myelin-associated glycoprotein, NOGO-A, and oligodendrocyte myelin glycoprotein are proteins produced by oligodendrocytes that surround the site of injury and affect myelin production and axon growth. Scientists found that by adding antibodies that bind to these proteins, they were able to block the effects of these proteins and allow axons to grow. For nerve regeneration to occur, we need three key ingredients: a scaffold or bridge, growth-promoting cells, and growth factors. These three elements, when combined together, can help bridge the gap of the injury to the spinal cord and allow axons to regenerate and grow across the injury site to make new connections, restoring function. Scaffolds are engineered mesh tubes or trellis-shaped structures. They can be made from several different types of materials, but are often made of a biodegradable polymer because they are easy to make, strong, and can dissolve in the body once their job is complete, in much the same way as stitches. The scaffolds are injected with growth-promoting cells and growth factors, often replacing those lost or damaged at the time of injury. The scaffolds are placed across the site of injury to act as a bridge that promotes nerve regeneration. Neurons are not the only cell type in the central nervous system. They need a little help from other cell types to replace myelin and ensure proper functioning. This is where Schwann cells, olfactory and sheathing cells, and enteroglia come in play. These glial cells, found in the peripheral nervous system, nasal cavity, and intestines, have the ability to remyelinate and regenerate spinal cord axons and help guide their growth to make functional connections. Neurotrophins like GDNF, NT3, BDNF, and NGF are protein growth factors that promote nerve growth and survival by blocking natural cell death. They can help regrow damaged neurons, remyelinate their axons, and help guide their growth to make functional connections. Neurotrophic factors act like vitamins for neurons to help them grow. They can also help encourage stem cells to form into neurons. In one study, Schwann cells were injected into a polymer scaffold and placed in the spinal cord of injured rats. Then neurotrophins were introduced, and 3 months after injection, the research team found nearly 5,000 nerve fibers growing throughout the length of the polymer scaffolds. In the next video, we'll talk about retraining using intact circuits to restore function.